guys, it's Miss Wisdom. I just want you to think back to a couple weeks ago when we discussed the cell cycle and mutation. What I'm going to do is I'm going to go through those processes again real quick to hopefully clear up any confusion that there might have been, and make sure we really got this down. So we're going to break it down. So what we did in our exploration, just to refresh your memory, is we considered a single cell and we tried to come up with the steps that that cell would need to undertake in order to divide into two identical cells. So some of the things that you guys thought of, you guys drew some stuff that looked kind of like that, with kind of a pinched off in the middle cell. You guys drew stuff that looked kind of like that, with a cell with like two nuclei. And these are all really good ideas. And we're going to go ahead and explore those ideas in more in depth. So the major regulators for the cell cycle are CDK and cyclin. And what those are just molecules that monitor the progress of the cell cycle and signal the cell to move on to the next step or not, and make sure that everything is proceeding as it should. So the cell receives a signal to divide. So that green stuff right there is just the cell receiving a signal to divide. So what can cause that signal is anything from like an injury or just a growing fetus, or anything really can signal the cell to divide. So our cell can't just divide straight in half. If we did that, we would end up with some little tiny cells. They'd only contain half the DNA that they're supposed to, and they really just wouldn't function properly. So we can't do that. So what we're going to do instead is we're going to enter the gap 1 phase, or G1 phase, which is basically where the cell just grows to make sure that we end up with the proper size of cell at the end. So our cell is going to grow. We've got our bigger cell there. And again, CDK and cyclin are monitoring this process. We're going to go through a checkpoint to ensure that the cell is the correct size. CDK and cyclin check that, and that works out, so we're good, and we can move on with the process. We've got our big cell there. The next thing we're going to do is we're going to go into the nucleus, and we're going to see what's going on in there. So we're going to kind of zoom in on the nucleus there. That red strand right there is going to be our DNA, and that's how the DNA is just in the cell during normal function. It's just a big long strand of code, basically. So in the next phase, we're going to copy that DNA. And what we're going to end up with is we're going to end up with 2 strands of DNA. And this is the synthesis phase. So that little blue circle there is a protein that's going to come through and read and copy the strand of DNA. So that's going to go through, it's going to read the strand, and then it's going to copy the DNA there. So the copied DNA is going to be identical to the DNA that we started out with. So those are the same exact DNA. Synthesis basically is just a fancy word for creation. So what we're basically doing is we're creating a second strand of DNA in this phase. Again, those strands are going to be identical. We're going to see our cell right there with a single strand go through the synthesis phase and end up with 2 strands. So our cell, which is big, it has 2 strands of DNA. We're going to now, CDK and cyclin are going to tell the cell to enter the gap 2 or G2 phase, which is basically just more growth. So the cell's going to get even bigger, and then CDK and cyclin are going to enter into checkpoint 2, where we check to make sure that the cell is the correct size again. We check that. And then we check to make sure that the DNA that we just copied is intact and correctly duplicated. So in G2, we check the DNA that we copied from the synthesis phase. So if anything went wrong, it would have been in the synthesis phase. G2 is just checking to make sure that it's right. So if everything checks off, CDK and cyclin are going to go ahead and signal the cell to move on into the process. And the next phase is going to be where we actually start splitting things. Mitosis, or M phase, is when the nucleus is going to split. So we're going to end up with something very similar to what you guys drew in our exploration there, with kind of a pinched off in the middle cell with two nuclei. So the way that we do this is we have our single strand of DNA, and in order to split that up between two different cells, what we're going to have to do is we're going to coil it up really, really tight. We're going to make it into like a tiny little spiral, and we're going to have our two strands of DNA there, and those are going to make two super tight coils of DNA that kind of come together in the middle like an X. And that X is called a chromosome. And the chromosome is basically, like I said, just a really tightly coiled packet of DNA so that we can actually put them on the two different sides of the cell. So we're going to go through the phases of mitosis really quickly. Those aren't super important for this, but just for your information. So we've got our cell there. We're going to first go through prophase, which is where we've got our nucleus broke down, the nuclear envelope broke down, and our strands of DNA condensed into those chromosomes there. Then the chromosomes are going to get lined up in the middle of the cell right there. The blue things are just the spindles that are attached to the chromosomes. And in anaphase, those spindles are going to pull the two strands apart from each other. So we end up with half of the DNA on one side, half of the DNA on the other side of the cell. And we've got those there in telophase. Telophase is kind of the process of those strands uncoiling from their chromosomes and becoming just the big long strands again. And then also the nuclear membrane forms again around that. So what we end up with here is our nuclei have split. Again, CDK and cyclin are regulating this process, and the next step is going to be cytokinesis, where the actual cell splits. And what we're going to end up with after that is two identical daughter cells with the same DNA in both, since we copied it. And so in summary, really quickly, we have our single cell. We go through the G1 phase. We grow to a bigger size. We go through the synthesis phase where we double our DNA. We go through the G2 phase where we again grow some more. And then we go through mitosis, or M phase, where we actually separate that duplicated DNA into the two sides. Then we do cytokinesis, where the cell actually splits into two, and we end up with those two identical daughter cells. Next, we're going to talk about some things that might go wrong in this process. We'll just go through each step by step. You can follow along on the screen there. Some things that might go wrong in the GAP1 phase is the cell doesn't grow to the proper size, in which case CDK and cyclin are just going to keep telling the cell to grow. In the synthesis phase, this is the major cause of mutation, which basically just means that when our blue circle went through and read our DNA, something went wrong and it didn't get copied properly, or the DNA got damaged in some way. So basically we're going to end up with two strands of DNA that are actually different. You can see there the green little portion is the portion of our DNA right there that is damaged or mutated in some way. So that is going to get picked up on in the GAP2 phase when we go through that checkpoint. However, if in the GAP2 phase we do not detect incorrect DNA synthesis, which is something that can go wrong in GAP2, then the cell is going to have the mutation. Generally speaking, the cell is going to notice the mutation, and CDK and cyclin are going to signal the cell to either attempt to fix the DNA, or if it can't be fixed, they will go ahead and just signal the cell to self-destruct, basically, or they're going to kill the cell. It's called apoptosis. So when this doesn't occur and the cell is able to continue on in the process, we end up with a cell that is different from the parent cell that we started out with. So this difference can be silent, which basically does nothing. It doesn't affect anything, any changes. Most mutations are going to be silent. Some mutations might actually be beneficial. This is actually the major cause of evolution and change in species, because some little proteins or anything might change ever so slightly and actually make the cell run more efficiently. And there are also mutations that affect the cell cycle, which we'll go into later, and they might affect things in the cell cycle like CDK and cyclin. And okay, so things that might go wrong in mitosis, the nuclei might not divide correctly. If you end up with an incorrect number of chromosomes there, like if the spindles didn't pull the two pieces of the chromosome apart, you might end up with one with the entire chromosome on one side and nothing on the other side, like in the picture. In which case that can cause many different diseases, particularly when developing fetuses. And then, let's see, cytokinesis. Again, you can wind up with the cell not dividing properly. You might end up with two nuclei in one cell and nothing in the other cell, in which case that's just going to break up and not function properly. So we're going to go back to talking about mutations and what happens if the mutation actually affects the cell cycle. So we're going to talk actually about cancer. So assuming the DNA has mutated and the cell did not detect the mutation in the checkpoint, and the mutation affects the ability of the cell to detect stop signals. So the cell is not receiving information to stop dividing. What's going to happen is the cell's just going to continuously divide rapidly. So we've got our single mutated cell there. We're going to end up we're going to end up dividing. Those are going to divide. Those are going to divide. We're going to keep dividing and dividing until we've got this huge mass of mutated cells that aren't functioning properly. And what we call that mass there is a tumor. And tumors can be non-cancerous, but they can also be cancerous. And obviously that's going to lead to illness. These cancers can actually be caused by a variety of things. We talked about on that second day when we talked about sunlight can cause mutations in our DNA. X-ray radiation exposure can cause mutations. We talked about the coal dust. We talked about a bunch of things that can potentially mutate our DNA. So we definitely want to try and limit exposure to that sort of thing, no mutagens like that, to lessen the possibility of causing these tumors, which may become cancerous. I hope that cleared up some things for you guys, and I hope you guys had an okay time with my lesson, and I wish you all the best of luck for the rest of your semester. Thank you.